So good morning, everyone. Thanks for coming this morning. We have a few just like housekeeping slides. Can you forward? Okay, just to remind you, all the primers are recorded, posted to YouTube. Any questions you ask will be in the video. You can use the Zoom chat if you want to ask your questions anonymously. We'll read them for you, no problem. Next week is a meeting-free week, so no primer, but the week after that we have the retreat. So there's no primer on the 5th, but there is one the following week, and I think it's Chris Casa. Okay. But today we have Dr. Peng Gao, who is an assistant professor at the Harvard Chan School of Public Health. Dr. Gao is in environmental health and exposomics. His research focuses on integrating chemical exposomics with multi-omic approaches to understand environmental contributions to chronic diseases. So this is another layer of data on top of genomics data. He pioneered precision environmental health monitoring through longitudinal exposome profiling and developed novel methods for spatial exposomics. His work spans from population-level studies to cellular resolution analysis, looking at how environmental exposures interact with genomic factors and disease etiology. So thank you, Dr. Gao, and we will hand it off to you.
Thank you so much for the introduction. Morning, everyone. Thank you so much for having me today. And as you can see, I'm the affiliate faculty here, so I am here a few times for attending seminar, but it's my first time people talk. It's a great pleasure. So I just want to ask a quick question. Have anyone heard the term of the exposomics or exposome before? Okay, maybe just one or two, three. Okay, great. Yeah, so I think that's worthy to give the talk because most of you have never heard it. And probably you will be curious why genomics need exposomics, right? Especially Broad Institute is so good at doing genomics research, but I will tell you more today. So the human phenotype is so complicated. It's not only driven by the genomics, it's also being impacted by the environmental factors. So by the name, you can see that exposome is the ohm of the exposures. Initially, people will think, okay, just include the chemical exposure, like the pesticides, flame retardant in this room, as well as some heavy metal. But however, right now the term is trying to expand it and then cover more different type of environmental agents, which could be important components to shape the human phenotype. So just based on the large cohort database, we can see that a paper published in 2019 showed that about, they kind of analyzed about 560 phenotypes and then find the genomics factor only contribute in average to like 30%. However, rest of the contribution are coming from the either GXE interaction or the exposome factors by themselves. So I already briefly introduced what is exposome. It's the collective term of all the exposures, which include super ambitious, trying to include all the external factors besides the genetic factors. So usually right now we are trying to categorize them into 4 domains. The first one is chemical, like the organic pollutants, inorganic pollutants, and biological, like the microbial exposure, virus exposure. The third one is physical, like the temperature, humidity, or radiation, etc., which some of them fall into the climate-related exposure. The last one is the psychosocial, which is also an important component and also overlooked historically. People may not think their mental stress or emotional expression or their relationship with a colleague may impact their biology, but actually there are a few studies already show it could be connected. So my lab has been focused on the chemical space alone for the past decade, and I will mainly only cover the chemical part during this talk. But I just want to emphasize that it's a much more broader term, not just only focused on chemical substance. So if we imagine like the exposome as a mountain, there is old Chinese poetry means that if we observe the same mountain from different angle, different height, you will see definitely different picture, right? So it's the same for the human exposome. because historically, conventional method can only focus on one single chemical or a single group of chemical when they doing like association or toxicology study. However, it's kind of lack the holistic or comprehensive view of all the exposure we are encountering in our daily life. Also, if we take the chemical agents as an example, after they get into human body, the situation will become much more complicated. Depends on the mixture you are exposed to. It will impact you at different layers, from the genomics, epigenomics, mitochondrial, endocrine system, as well as the cell-cell interaction. Microbiome, for sure, we see the microbiome biotransform certain type of xenobiotics and increase their toxicities, neurotoxicities as well, like the traditional one, lead, we know, and many pesticides can be neurotoxic, especially for vulnerable population like the child and elder. And the oxidative stress and the inflammation are the really common first effect it will cause when whatever type of the xenobiotic get into your body. So it's a really systematic levels impact. That's the reason my lab have been focused on integrating the exposome to the multi-omics layer. So there are quite a few studies already show that the exposome mixture effect do exist. Even going back to the reference publishing more than 10 years ago, they find that inorganic pollutant and organic pollutant can interact with each other and then promote the toxicity for each other. We call a synergenic effect or more than additive effect. Also, compared with the chemical assay, with the biochemistry assay, we found that usually the bioassay can see much more toxicity effect compared with the chemical alone, which means that the chemical interact with each other and then contribute to more toxicity than just when you just add them up. Recent year, one published last year, the other one published in 2022. We can see more and more exposome or exposome-related paper published in high-profile journal. These two are two examples, both focus on neurotoxicity. The one on the left are focused on the mostly the neurotoxic chemicals, and they find that the single individual stressors do not contribute to overall toxicity when they just add them up. So which imply the more than additive effect or the synergenic effect I mentioned. Another one is that they monitoring the cohort blood and then prioritize a few type of EDC, which are the endocrine disruptor chemicals widely present in the environment. And based on the biomonitoring dose, they exposed to both in vitro and in vivo model, and they do find the early detection of the biomarker related to a language delay in the children, which both imply that the exposome mixture effect matters in the real world setting. So my lab, as I mentioned, focus on a chemical part for a long time, and we do both environmental monitoring as well as the biological monitoring. Usually we just take either environmental samples or the biospecimen from a different cohort and then do the mass spectrometry analysis. The reason for that is we are trying to meet in the middle, because if we see the whole pathway of how certain exposure can impact to the disease, there are pathway taking the benzoate pyrene here, which is a highly pro-carcinogen, usually coming from the incomplete combustion byproduct, such as the vehicle industry, barbecue as well. So after they get into human body, the enzyme will transfer them into a carcinogen, which showing here, and then being the adduct of the DNA, and then leading to cancer cell, eventually may lead to cancer or tumor. So that's the reason we do both monitoring and trying to get a more comprehensive pictures. So also after the chemical get into our body, they can interact with the different layers, as I mentioned, and that's the reason we are trying to integrate the multi-omics profile. First of all, I just want to give a brief introduction on what we have been done, focus on the population level. So my lab have been focused on applying exposomics approach to study the disease etiology at usually 3 level population, individual, and cellular level. So population level, usually we just gain the access from publicly available large cohort database. For example, UK Biobank, Genomics should know that because there are so many GWAS studies have been using that database. In US, we also have the All of Us. And then in the Europe, there are some certain smaller cohort specifically designed for studying exposome. In fact, for example, the Helix cohort is designed to study how the prenatal maternal exposure could lead to the health outcome of their child. And my lab have been also focused on the how to use the exposomics approach to study the environmental justice issue as well as the health disparity issue. If you look at the epidemiology data set, you can see there are huge disparity in terms of one disease, even within the same city. So my PhD work, which is a long time ago, already showed that the different type of environmental pollutant, they are existing in the same city disproportionately. So their concentration are significantly different, depends on where you are residing or work. So it also implies an environmental justice issue. Another challenging part of studying the exposome compared with the genomics derived omics is because it's super diverse. Because if we talk about genome, the base pair, we only have ATGC and SNPs. We usually just know them. But for the chemical part, even based on the current database, even we only look at the synthetic as well as the natural organic chemical, there are already more than 2 billion. It doesn't mean that we are exposed to 2 billion chemicals simultaneously every day. However, at least we do have thousands of them. And most of them are unknown. We don't know what they are, but they present, and we are inhale or thermal exposure to them. So also their abundant longitudinal changing. Really depends on your time and space. For example, these rooms are flame retardants released from the furniture. Their concentration are different from that room. So maybe you will have higher exposure in this room and then lower in that room. Also your home, as well as the transportation method will all impact the how diverse and how the abundance change. As I mentioned, it's dynamic as well. So it depends on your time and location and personal lifestyle. So I just briefly mentioned that my lab have been used in the mass spectrometry, especially high resolution mass spectrometry, to study the chemical part of the exposome. It's already been widely used in the metabolomics and lipidomics field because they are all small molecule, and the proteomics as well. I mean, not the antibody-based one, but my lab have been focused on to fine-tuning the pipeline and the protocol to specifically target and capture the xenobiotic part, not the endogenous part, although we can do both capture in a single run. But historically, most of the small molecule database are focused on the endogenous molecule. They don't care about the xenobiotic we exposed to at all. So my lab have been focused on both the targeted approach as well as the non-targeted approach. The first one is the hypothesis-driven methodology. For example, you may think, okay, neural disorder could be related to lead exposure. So we can just target the analyze lead or certain type of pesticide. For non-targeted, we just get a sample, usually blood, biospecimen or other type of biospecimen from the cohort, disease cohort, and then do simple case control comparison. And we don't need a specific hypothesis in hand because we are able to screen all type of small molecule present in the sample and then see what type of the small molecule could be related to the disease risk or progression. Besides the small molecule, my lab right now are also focused on the micro and nanoplastic, which is a hot topic recent years. The reason is that is because we don't know the health effect of them. We just know they present in our body. And also the concentration are not really clear. So my lab right now is trying to develop a robust, reliable protocol and full pipeline to analyze the micro and nanoplastic that in the blood samples, so we can apply to the human cohort and then to see whether it could be associated with any type of a disease. Also, the plastic by themselves, similar to the particular matter present in the air, they are acting as a vector. So they are a carrier of different type of persistent organic pollutant as well. So whenever they get into human body, they will take all this type of complex mixture chemical cocktail inside our body as well. So we don't know how they interact with each other either.
Can I ask a naive question?
Sure.
Regarding microplastics, I mean, obviously there's a lot unknown, but can't someone make animal models and expose animals to microplastics in a chronic way and understand how they affect the body that way? I mean, I'm sure there's research, but what do those types of studies show?
Yeah, I think it's totally feasible in the toxicology field. People use a different animal model exposed to certain type of plastic and different size, maybe acute or chronic exposure. However, most of them, I believe they are like the pure plastic, which is not reflect to the real world plastic unless they take the sample from the real environment, right? So that's one of the limitations. The other is that I think the dose they are using usually is much higher than what we really exposed to in the daily life. And we all know that the biology of the mouse and the human are different as well. Even inside the human population, we can see significant disparities because of their genetic susceptibility. Because based on the genetic makeup, even we exposed to the same dose of the same gluten, you may have an effect, but I don't, or reverse. It's because our genetic sensitivity are different. So it's a really complicated question, but for sure that's feasible. We can expose to whatever to animal model, but it could be helpful or may not be helpful to reflect back to the human real world case. So the other level I mentioned is to study the exposome at the personal level. We do find that even we have the cohort in San Francisco Bay Area, we deploy the personal level exposome monitor on their body. We found that their physical, biological, and chemical profile are significantly different. However, historically, in the environmental health field, we usually consider the participant in the same city, they have really similar exposure profile. And recently, we already have many evidence prove that it's wrong. So exposome is a really personal thing, and each individual can have really significant different exposome cloud, even they live in the same city or even in the same neighborhood. So that's the reason I came up with this paper, actually just published a few weeks ago as a single author in the new Nature journal called Nature Health, more focused on the population health rather than compared with Nature Medicine, more focused on the clinical implication. In this perspective paper, I just introduced how complex the airborne exposome could be. It's considered different type of thousands of organic, chemical, tens of inorganic, as well as even millions of microbials, and we are exposed to them simultaneously. And the traditional, for example, based on EPA regulation, they only focus on 6 criteria air pollutants, and you can see it's just a really tiny portion of the airborne exposome. So we really need some new framework or technology to study, for example, the individual exposome mixture model and the personal level exposure assessment, as well as the biological mechanism based on the system biology or multi-omics approach. And this slide shows some examples. Certain type of health outcome could be better understand based on the exposome mix approach, and then to see how the exposure mixture could lead to the risk or progression of all these type of disease, including neural disorders, cancers, cardiometabolic disorders, as well as immune-related disease. At personal level, how we check the airborne exposome? Usually there are 2 ways. The first way is using a passive sampler. The widely used one is the silicon wristband, because silicon is a really good material to absorb the SVOC that present in the environment, which is the semi-volatile organic compounds. So the SVOC are the major group of in terms of the organic part of the airborne exposome. So we in this study, we also developed another passive sampler which aim to capture the short chain PFAS. PFAS is a kind of emerging contaminants which they consider as a forever chemical. It may not be true, but it's persistent for sure. And the health effect is not really clear right now. And the traditional study only focused on long chain that present in the drinking water. However, we also find a higher abundance of the short chain, which are the alternative of the long chain. They are widely present in the air. Another approach is to using the active sampler. Active sampler do have a battery, have a pump, but definitely the size is much larger. But we are trying to improve it through different generation, like iPhone 1 to 3. Right now, we already end up with the generation 3, and you can see the performance being keep increasing. We are for when we deploy to the participant, we think the things matter usually more is like the battery life. So we are trying to increase certain type of performance of the device. It's a collaboration project with my previous colleague at Stanford. And the advantage of the active sampler is that we can customize different type of filter inside the device. So when I was there, we kind of integrate 3 different type of filters in the device. The first one is to capture the organic chemical. Second one is to capture the inorganic chemical. The third one is to capture the microbials communities. So by using that way, we can gather a much more comprehensive profile compared with the silicon widthband we use which only focus on organic part. However, active sampler is much more expensive compared with the silicon widthband, which is not doable to apply in the large cohort. My lab is not a traditional lab. We are really half and half. Half of my training are focused on wet lab, the other half focused on dry lab. The reason is because the field is too emerging. We don't have any tools or pipeline or the package that can handle the exposome data set. So we also focus on to kind of develop some new methodology to do with the complicated high-dimensional data set we generated. For example, we just published a new pipeline package called TidyMass2 in Nature Communication a few days ago. And also my current PhD students are focused on developing a machine learning algorithm to study and predict the unknown mass spectrometry features that we have been generated. Here is an example how we use the Exposomics methodology to check the exposome profile in the individual level longitudinally. So in genomics field, I know usually people don't care about the longitudinal profile, but as you can tell, exposome is really important to check them longitudinally because they keep changing. So we use the active sampler here, and then we only have 2 filters at that time placed in the device. One is to capture the biological part, which is the shotgun metagenomics NovaSeq. The other is using the orbital mass spec to do the small molecule non-targeted analysis. And based on the result, you can see there is a significant change in the middle of here in terms of chemical exposure. And then we go back to the metadata. We found the participants start to traveling. And you can see how traveling can impact the personal level exposure dramatically. And also the biological part, you can see when before the travel, the type of fungi which represented by the purple part is the dominant species. However, whenever they start to travel, they just no longer become dominant. And also we keep measuring the personal level environmental factors, general environmental factors like temperature, humidity, atmospheric pressure, etc. So we can see it's feasible to check the participant longitudinal exposome profile at the individual level. As I mentioned, because after the exposome substance get into human body, they can interact with different layers inside of the human body, from genomics to epi to transcriptomics, proteomics, and proteome and then metabolome. So different layers may have different interaction with the exposome xenobiotics. That's just about the chemical part, right? Because if the microbial get into human body, that would be another story. But even only look at the chemical part, we can see they have a really complex network, even just based on the statistical association. We found nearly 9,000 significant association when we correlated the chemical biological part with, as well as the general environmental exposure with the different internal multi-omics layer. And here are some specific detailed results from different one-to-one integration. This one is focused on the exposome correlated with the gut bacteria microbiome profile. Inner cycle is the bacteria, outside is the exposome substance. We found there are like 6 top high degree bacteria here. And then there is an interesting pattern is that besides the rosemary, which is the bacteria I show here, the pattern is that most of the exposome substance, they are positive correlated, negative correlated with chemical substance, but positive correlated with the airborne microbial. However, the rest of 5 have the reverse pattern. We don't know the reason for that, but it's just interesting to see this pattern within the top 6 high degree bacteria. And when we compare a different site microbiome and then correlate it with the airborne exposome, we can see to different extents they are also associated with the exposome substance as well, which makes sense because, for example, the external part like the skin, as well as the nasal microbiome are more easily being impacted by the airborne exposome, but the internal part are not. This is the detailed result from the exposome correlated with the proteome, as well as exposome correlated with the metabolome. It's a complex figure, so because we also did a downstream analysis trying to see how the pathways like the metabolic pathway as well as signaling pathway could correlate with the exposome substance. But the take-home message is that based on the proteomics analysis, we found that the most of the top significant pathway or proteins are related to the immune system. For the metabolome analysis, we find the kidney and liver function as well as some metabolism related to oxidative stress, amino fatty acid, bile acid metabolism. They are significantly correlated with the airborne substance. We also did a cross-validation in this study because usually multi-omics may derive a contradictory result when you correlate different layer of omics. However, in this study, we found a pretty consistent result because when we see the cytokine panel results compared with the proteomics result, they are consistent, kind of validate each other because the top one are pro-inflammatory related cytokine. And when we see the blood test result correlated with exposome, they are kind of validate the metabolomics correlation result because we found that the top one are related to the kidney or liver functions. And whenever we see some, we know in the clinic, they do have the reference range, right, for the blood test. And whenever we detect some values outside the reference range, we also see the significant change in terms of their exposome cloud substance. The last part I mentioned, we focus on the cellular part. So we do use different in vitro and in vivo models to expose them to environmental mixtures that we collect or derive from the real environmental samples. Here, for example, we use the human oral epithelial cell line to expose to the mixture of a heavy metal, and mixture of heavy metal are kind of collected from the real site, which is the contaminant industrial sites. And we also use in vivo model like zebrafish. For example, in this study, we investigate how the disinfection byproducts that present the drinking water treatment sites, and then how they can impact the neurodevelopment in the zebrafish. This specific study using a multi-omics approach to study how the environmental relevant high arsenic exposure could relate to the molecular change by using both cell line as well as the animal model. Because we did use the multi-omics approach, and then for example transcriptomics analysis as well as the metabolomics analysis, we find that these environmental relevant arsenic exposure could induce a change in terms of ferritosis. in both models, which we believe is the there are some papers also published a similar result, but it is one of the first of publishing this type of molecular change induced by the arsenic exposure. My lab currently also really interesting at the cellular, multicellular, or even single cellular levels exposure, because we know there are heterogeneity depends on cell type. Some of them are sensitivity to this type of gluten, and some other type are sensitive to other type. So we want to see how the environmental carcinogen or pro-carcinogen distribute, for example, at a tissue by using the imaging mass spec. We can also use the cell type-based exposomics methodologies similar to the single cell sequencing technology to see the concentration difference in terms of different cell types. So here is an example of you using the imaging mass spec. There are different type of imaging mass spec in the market. However, my lab have been used the Waters DAISY source because we don't need a specific sample prep for we just need to slide. And the sample can be reused for sequencing even after we scanning the small molecule by the DAISY high resolution mass spec. But you can see, for example, here we use the human lung tissue sample, and we can see certain type of xenobiology are distributed differently across the tissue. So as I mentioned,
can I ask a quick question online?
Yeah.
Could you briefly touch on throughout those analyses where you're getting your samples and approximately how many samples you're using in them? I think it's specifically for the personal profiling.
Personal profiling. The device one?
Yeah.
Okay.
Yeah. Yep.
What's your question? Sorry.
Where you're getting the samples and approximately how many samples you have, especially to build like the reference that you're comparing metrics to.
Yeah, we get a sample from the real participant, so we just deploy the sampler to specific cohort. Depends on the study. Sometimes study they only have the patient, but some of them also have the healthy control. In terms of sample size, it really depends. It's super hard. For example, the asthma cohort I've been working on, I will show you some preliminary data later. But we spent 3 years, only get 50 participants return the sampler. So that's the reason barely are people doing this research because it's super time and money consuming. But I do believe it's important because it really reflects your real world exposure.
Thank you.
There will be another problem because if you are familiar with the epidemiology field, they really care about the sample size in terms of the statistical power. So sometimes they will challenge on that, especially the omics data set that are high dimensional, even require more sample size. However, again, as I mentioned, it's super time and funding consuming. So in the future, we just want to use this type of methodology based on the pilot study we have developed to apply into larger pool. So I think the last part is the perspective part. I just want to emphasize that the exposomics field is really transdisciplinary field. We do need to improve from different fields like chemistry, biology, environmental science, and data science, super important. And right now we're using different type of machine learning algorithm to deal with or predict certain type of things we care about. Also in the geospatial field, they are trying to profile the high dimensional exposure cloud at a larger level like neighborhood or even city. So it's also an important component as well. So that's all I have today. I think because I'm right on time, but I think I still can take 1 or 2 questions. Thank you for your attention. And again, I just want to acknowledge all the collaborators and the funding agency. Thank you so much.